Thank you so much, Justin. I appreciate the welcome and the invitation from Nanopore, Oxford Nanopore Technologies to be here. Let's get going. So in New York, Wadsworth Center, we're the state public health laboratory. We have a large population of about 19 million. We do work to rapidly determine the source of outbreaks. We do reference level testing to support our clinical laboratories in the state. We do national surveillance and also a lot of national testing around TB at the moment. And this is just showing you some of the focus work we do, just to give you a feel for our role. And then we've been fortunate to do a lot of assay development focused on additional characterization as well as emerging infectious diseases. And so a lot of that work is around antimicrobial resistance and a good amount of work around TB, which are what I'm going to talk to you mostly about today. And so about 5 years ago, when we started to think about how we could fit nanopore sequencing into our work that would really impact our testing, we first thought of the long-read sequencing, of course, and with the AR gene detection we do and plasmid analysis for understanding transmission across the whole northeast of the U.S., we came up with a lot of strategies and applications where we could apply that. I'll mention a few of those to you. And then for Mycobacteria, both for the rapid drug resistance detection of TB cases. So we started that work in about 2021. We brought on a clinical assay in April of 2023. So we've had a few years of using that in practice, and it's been working really well for us. And then I also just want to mention at the end some NTM detection, because that's a very complicated testing algorithm for us. And so first on the nanopore sequencing of antibiotic-resistant bacteria. So we're of course interested in characterizing mobile genetic elements that carry novel or uncommon resistance genes. And we've been able to identify some that were novel to us in the US and to our region that were helpful for investigations. And then the genomic context of those genes, whether they are on the chromosome or on plasmids, to really help us apply those and reducing transmission in our healthcare facilities. And then we have done some investigations on the possible plasmid transfer events associated with healthcare facility investigations. And just on the few of these slides, I have some from our group. About half of the people on this are bioinformatics experts or research scientists in our group, and then wet lab experts. And so I'm just trying to capture that this work is done as part of a large team. And so I think that we're all familiar with some of the reasons for using the long read, especially the Oxford Nanopore methods to characterize those plasmids. And when we look at that characterization, when we're just using short reads, we could not tell where the genes were located. When we did our analysis, we saw this sort of a jumble, and it wasn't clear whether we were dealing with plasmids or these were on the chromosome. But by doing some hybrid analysis initially, and we're looking forward to seeing if we could bring this to only long-read analysis in the future, but it's really been helpful to us to understand. And so one investigation I just wanted to mention that we had KPC producing bacteria at 2 healthcare facilities, and this is in one of the states in our region that has a very low level of carbapenemase producing bacteria identified. We believe that there was a plasmid carrying the block KPC2 gene that moved between Citrobacter and Klebsiella pneumoniae and E. coli. And so in this investigation, and this is Maine, we had a number of patients for which we had isolates, and we have an epi curve that helped us understand that some of them crossed between the same healthcare facilities over time. And so we did a lot of work doing both short read and long read analyses and some plasmid analysis to help us really understand that this really could have happened. It's not an absolute because the plasmids identified were not completely unique, but because this was such a low level of infection and of transmission in these facilities, we felt that this was substantial evidence, and we were able to publish that work. Another investigation, and so vancomycin-resistant Staph aureus, we've only really had one documented case in New York that I'm aware of in 2004, but it was a very interesting case where we had a person who was Versa positive for about four and a half months in a long-term care facility before she passed away. And we were able to receive many specimens over this period where we were identified and isolated Versa strains, vancomycin-resistant enterococci strains, as well as both methicillin-resistant and methicillin-sensitive strains. And we used long and short read analysis to better understand what was happening in this person who had multiple indwelling catheters over time, just so we could try to understand this for the future and future investigations. And so the Versa isolate, as you would guess by looking at these strains, emerged from the transfer of the VAN-A gene from the VRE to a MRSA strain, but also to an MSSA strain. The plasmid in some of the isolates did integrate into the chromosome via homologous recombination, and we were able to just put some of this work together and publish this as well to try to just have this information out there. We weren't aware at the time if other examples of the VAN-A gene moving into the chromosome had been observed, so we wanted to publish that. And then lastly, I just want to mention we had a very large outbreak over the last several years with a Pseudomonas aeruginosa strain that was found in many patients across the U.S. You can see the map here on the bottom right. We had quite a few in New York, and our first isolate came to us from the state of Connecticut. When we did the whole genome sequencing in this investigation, we determined it to be a very unusual strain, one we hadn't seen before, with both BLA-VIM80 and BLA-GES9. And the sequence type 1203. We did quite a bit of work to get additional isolates from our region to do more testing, and we're able to do long read sequencing as well to better understand that isolate. Submitted all of our isolates to NCBI in the US very quickly so that the CDC could use that information to track down the sources, and there was a lot of analysis done. This was published, and it was a very interesting study for us to take part in and utilize these great NGS tools. And so now I'm going to shift gears and talk about TB, Mycobacterium tuberculosis complex. And so I think we all know TB is a really important infection, especially globally. New York in the US has the third largest number of cases, and about 20% of these have drug resistance, and this is increasing. I was just looking at the 2024 data, and while we were down to about 700 cases for our state in a few years ago, those rates have gone up 30% each year, and we're above 1,000, which we haven't been in a very long time in New York. So because of the pandemic, as well as some migration, we're dealing with a lot of additional cases. And so we have been working for a long time on methods. So we've been doing clinical whole genome sequencing since 2016, but we really wanted to be able to do that direct specimen work. And so, like I said, we brought on the whole genome sequencing. You can see this here. In 2018, we stopped doing typical phenotypic susceptibility testing for those that were pan-susceptible isolates predicted by our whole genome sequencing testing. And we created this test that I'm going to tell you about. And just to give you a feel for, where we're located in the Northeast, you can see we're kind of here in New York, very close to a lot of the states, but providing this work. And those cases I mentioned make up the approximately 8,000 cases in the US. And in terms of our laboratory, we don't get all specimens from suspect TB cases. We get a highly priority group of those specimens, so we can really focus our work. And in 2023, just as an example, we received almost 4,000 samples. 726 of them were TB cases. More than 500 of those were isolates. Over 100 were primary specimens, as well as recultured isolates made up a small amount. 96% of those were Mycobacterium tuberculosis, and 14% of those were drug resistant. And so to focus on those specimens, and this has gone up, and I mentioned we're doing some national testing in the US at the moment, we brought on a test using Oxford Nanopore, Nanopore sequencing, and we're using DNA extraction with an automated EZ1 test. We have we started this development before there were available tests, but we developed this and also so that it could use the same sort of pipeline analysis to be consistent with our whole genome sequencing assay. But we have a targeted multiplex approach with 2 different tubes, and I'll mention that in a moment. We're looking at 13 targets and 9 antimicrobials. We're doing manual library prep. We're using the GridION and doing sequencing for less than 2 hours. We're washing our flow cells up to 3 times, depending on the poor health. and we're using an in-house pipeline for that analysis. This testing on the specimens is offering more information than our previous pyrosequencing assay did, and we're finding the results to be very concordant with our whole genome sequencing test, which is also occurring on every case in New York. And just to show you how that assay is set up, these two pools, there's a lot of work that went into optimizing and validating these pools and the specifics of how they're combined and barcoded. We're using native barcoding for this, but we're excited about maybe the new topo kits for the future, where the rapid barcoding isn't as effective for this assay. We're also not able to do our bead beating that we would typically do for other TB testing because of the size of these fragments. But this just gives you a feel for the targets involved and how that's set up for us. And this is a little bit of our testing to date. You can see we've tested a number of different specimens since we brought this on. We've, of course, seen many pan-susceptible strains, but also some resistant, including 12 multi-drug resistant and 1 extensively drug resistant, which I'll tell you about. Overall, we're very excited that we really are truly getting some more information sooner. We have an average of 15-day improvement for primary specimens. We're also occasionally using this test for isolates that are contaminated or mixed with MAC or another NTM, and we are seeing a 43-day improvement for that mixed specimen testing. And we can do this testing in less than 24 hours if needed, and I'll show you one example of that. Our costs, it depends how many we're running, but are generally less than $100 a sample. And compared to our whole genome sequencing, that takes about 14 to 21 day turnaround time from specimen receipt, although we can do that rather effectively now at $65 a sample, but now we can do both of those tests for each sample for what it used to cost us to do one whole genome sequencing in the past. And this is just a picture of our testing algorithm and just kind of focused on, I mentioned the targeted NGS that I talked about is here. We also have a more sensitive one we're using when we see lower burden of TB in a sample when the CT is above 30 with our PCR assay. And we have another test that is coming that will be a reflex test for the newer drugs. And so the couple of examples I wanted to tell you about, in this one case, we had a 32-week pregnant woman. She was rifampin-resistant TB on the Cepheid test in a sputum sample, and this was sent to us at Wadsworth Center. We received the sample on a Friday, and on that day, the next day, we were able to have a PCR detection. And on day 5, we had a targeted NGS was run, and we could see that this was a highly resistant strain. We saw these mutations, and we had this interpretation. But the pyrazinamide, we didn't have amplification of that target, which does sometimes happen with some of our targets. And we saw an unknown mutation for ethionamide. But we move along. We had a culture positive on day 13, and we finally had a whole genome sequencing available on day 26. You can see the timeline for that. And the DST, some of the DST was available at 84 days. Some of it was never available because of the slow-growing nature of this particular strain. And so this for us was a milestone that we could use this test, a targeted nanopore test, to detect an XDR directly from the specimen in 5 days from specimen receipt. We had another case of a patient where there was a rifampin-resistant MTPC detected in sputum again. It was sent to us for testing, and this was an example where we could very quickly determine it was a false rifampin-resistant gene expert result caused by a silent RPOB mutation. And so we're able to test that, report that out, and make sure the person was on the correct antimicrobial drug regimen. And in the last case, we had stat testing, and this is the case we did in less than 24 hours. A 12-month-old that was very sick, had a TB positive relative. We had a CSF sample that grew MTBC positive isolate. This was sent to us at Wadsworth Center. And we were able to determine with the targeted NGS result in less than 24 hours that this was a pan-susceptible strain in terms of treatment. And then just finally, I want to mention that we do quite a bit of work for NTMs as part of our testing algorithm. We have some real-time PCRs you can see highlighted in yellow. Our initial PCR is MAC and MTBC. We have a separate one for abscesses. We do Molotov mass spec. We do Sanger sequencing and whole genome sequencing in some cases. So with our experience doing targeted NGS with the Nanopore test, we thought we could develop a test that would allow us to do NTMID with a TNGS test in about 2 days with just one test and replace all of those additional tests. And this is something that we are working on. So we've developed a workflow, we've developed a pipeline and a strategy, and we're about to move into some side-by-side clinical testing. So we're very excited that we may be able to do this and add it to our other TNGS work. And then finally, we're doing some work with Neisseria, meningitis, and gonorrhea for antimicrobial resistance. And so we're still working through some of the details, but we're very excited that this may have some rapid ability to look at many genes and help us kind of move to determine antimicrobial resistance and susceptibility from a targeted NGS approach. And so I'm going to end there. I thank you all for your attention. I want to thank the staff that I pictured throughout, our cores that are a part of the Wadsworth Center, our CDC funding, contracts that we have. A huge thank you to Oxford Nanopore for all the support developing the tests all along the way. And also Illumina sequencing is a backbone to the work we do at Wadsworth Center, and it's been very important in those investigations. So thank you very much.